Hi, welcome to GS 101 Laboratory. This is going to be a little different kind of a laboratory experience for you today. For one reason, we're doing intertidal diversity. And the two major reasons why we don't have the whole class over here. One, 800 students logistically would be almost impossible to handle, particularly for a beneficial educational experience. And two, that many feet would be very destructive in this particularly sensitive environmental area. So what we've chosen to do is I'm going to come over here and experience the tide pools in person, and you're going to kind of vicariously drift along with me there in the laboratory. And you have some things in your packet that you probably should be getting out now. One is the diagram of the intertidal areas, and the other one that we'll use a little bit later is that chart of different organisms. So if you have those on the lab table, we'll just kind of move right along. I'm John Lyford. I've already introduced myself in lecture this week. So we'll look at three different aspects of the intertidal zone. One, we're going to look at what causes the zonation of life in this zone where the tides move in and out twice each day. Two, we'll look at the diversity of life that lives in each of those zones. And three, we'll look at some of the dynamics of how they can exist over here and some of the feeding strategies that they have. You know, on the west coast of North America, the tides go out twice each day. So we have two low tides and two high tides, each 24-hour period, or approximately 24-hour period. Now, if you examine the low tides, and for a year's period, you average the lowest of the lows. Now, those two low tides are not equally low. So if you average the lowest of the lows, that would be, by definition, a zero tide. And anything less than that is a minus tide. That is that the water's out so that we can examine the intertidal life. You might have wondered what a minus tide was when you look at the tide tables. Now, there's several things that cause this zonation. But first, let's look at this rock that's right behind me. This rock is facing you, and that's to the east. So behind me is the north side of this rock, and in front of me is the south side. Now let's look at some organisms that we're going to see in a little bit more detail later that are growing on this rock. Up here at the highest level, we find some little organisms that look like little hats stuck onto the rock. These are limpets. They're in the phylum Mollusca, and I'll talk more about those later. And as we come down, we find some rockweed that's growing here. And further down, in this area of the rock, we find some what are called gooseneck barnacles. You can see that the fleshy part of the neck is quite neck-like, and so they're called gooseneck barnacles. And then even farther down, often associated right with the gooseneck barnacles, are these mussels. Now what I'd like to do, we now know limpets, gooseneck barnacles, and mussels. Let's just use this meter stick and see if there's any difference in how high they grow, depending upon which aspect of this rock that we're looking at, whether we're looking at east or south or behind me north. Let's walk around to the south side here and examine this first. Okay, now let's keep in mind where we were on those mussels particularly. I've got my thumb here on the meter stick where they were on the east side, and now if you look down in here, you see they're considerably lower here on the south side. And the same thing holds true for the barnacles that we saw, and up here the rockweed and the limpets. So now let's walk around on the north side and get the same sort of aspect and see if we can see a difference in level over there. Okay, now I'm walking around here to the north aspect of this rock. Remember where the mussels were growing over there in the south aspect, way down here. And now let's look as we go up here on this north side. Here are still mussels. Here are still mussels at the end of the meter stick. And here they are way up in here, more than a meter high here on the north side. Gooseneck barnacles and mussels, and the limpets are even higher as we move on up the rock. They're found up this high. Now the question is, why is this? And it has to do with the tilt of the Earth at 23.5 degrees with respect to the sun. And I'll ask you this question and let you figure it out by reading the chapter and listening to Mr. King's lecture on the biosphere. If we stood here 365 days a year, right here at this spot. Would the sun ever appear directly over my head? And the answer to that question will give you some clue as to why the zonation is higher on this north side than it is on the south side. Let's examine what happens on the south side if there's a little protection. Now I'm on the south side of this rock again, and remember over there on that boulder on the south side, these gooseneck barnacles and mussels were only growing about that high from where the water would come in. But now let's look here, also on the south side. As we move up here, we find that in this crevice, they're growing up at least 2 or 3 meters up into that crevice. And the reason for that is that the organisms are experiencing a microenvironment in that crevice that's entirely different than the environment that I'm experiencing out here, just feet away. And so these sorts of phenomenon, these physical and biological aspects of this zonation, are what causes these organisms to be successful in their adaptations at different tide levels. Now, one thing that we'll notice today is that these zones, if it's a broad, flat plain, like you saw in the introductory clip, these zones are drawn out accordion-like, and so they're very broad. But as the face of the cliff gets more vertical, those zones are collapsed, and they're into very narrow bands. And you saw a comparison just as this program started. Before I go in to talk about in more detail the diversity of life found out here, let's examine for just a minute some of the causes of the zonation that we see in this intertidal area. One, I think it's pretty obvious that the water going in and out, the physical factors. For instance, when the water goes out, it's a whole different life out here. All the organisms have to close up, and they have to contend with the drying effect of that dry air. They also have to have some way to hold themselves in place, so they have to have some sort of a hold-fast mechanism to keep themselves from being washed out or banged around on these rocks. And second of all, they have to deal with the light, the changing in the light, particularly the plants, and they have to deal in the wintertime with freezing temperatures, and they have to deal also primarily in the wintertime with a lot of rain. So they're marine organisms and they're being diluted. But not all of the factors out here of the zonation are physical factors. It has been found through a lot of real diligent research that many of the factors are biological. That is, the organisms are competing for space, and some are eating others, the predation. And so if a predator has a very good adaptations for a particular zone, and it has a favorite meal there, the organism that it's its prey will not be found in great numbers there, but may be found in an area that will not accommodate the predator. So it's quite complex. Well, now let's go into some diversity, and I'll just do this kingdom by kingdom and phylum by phylum, pretty much as it is in the appendix of your book and also in the chapter that you were assigned. Kingdom Protista. The trouble is with that one is they're all microscopic. But I brought along a little fine mesh plankton net here. And if I throw this just a few times out here to concentrate whatever might be in this water, we'll see if we can get something in this little flask hanging off the end. This is brilliantly clear water. If you just examine it like I am just with my unaided eyes, I can't see a thing floating in there. Looks like distilled water. Of course, it's salt water. But when I drag this net in here, much like an oceanographic vessel would drag a net through the open ocean, and I examine what I capture, in this flask at the bottom, I see a myriad of activity. Organisms that are swimming around that are obviously animals, and then there's a slight tint of green in here, kind of a brownish green. And if I looked at those with the microscope, what I'd find out here are primarily diatoms in the group Chrysophyta, and the dinoflagellates were in the group Pyrophyta. Although the kingdom Protista is well represented in this plankton sample, the members are microscopic, and we can only see them with the unaided eye as slightly coloring the water. The organisms we do see swimming about are not protists, but are in the phylum Arthropoda, class Crustacea, and are generally referred to as zooplankton. Your textbook discusses the insects, another class of arthropods, at some length, and the insects are important, terrestrial creatures. However, the insects, with few exceptions, are not found in the oceans. The role insects play on land is played by crustaceans in the oceans. Truly, the crustaceans are the insects of the sea. So these are the protists, and these are the organisms that many of the other organisms that I'm going to examine now will feed upon with various kinds of filtering mechanisms. Unfortunately, that's about all we can do today with the kingdom Protista. They're all microscopic. And I'd like to start now with the kingdom Plantae. There's only one plant down here, one species, that actually is in the phylum or division in the plant kingdom, Anthrophyta, the flowering plants. And we see it right here. It's called surf grass. In fact, it looks very much like grass, and it's the only plant that we'll find in the intertidal zone that is in the flowering group. All the rest of the plant divisions, are non-flowering, that is, generally speaking, are called algae. And they're divided into three major groups, pretty much according to what kinds of pigments that they have. The first one, the Chlorophyta, are bright green, like this sea lettuce that I have here. In fact, this plant that I just picked up only has one cell layer in thickness, although it looks like it might be longer than that. If you looked at it with the microscope, it's quite a fragile thing. All the Chlorophyta have chlorophyll A, that's why they're green. In fact, all the algae have chlorophyll A. But in some of the other groups, the chlorophyll green is masked by other pigments. So this is an example of a Chlorophyta. And here's another species of Chlorophyta down here. Incidentally, on your little charts, on your lab tables there, we're standing right here now about the mid-tidal zone, so you can kind of mark these different algal species down as mid-tidal. The other two groups of marine algae are the reds, which I have right here, and unfortunately they're not all bright red in appearance. In fact, they're kind of maroon color. Here's one species, and here's another species over here. These are in the division Rhodophyta. They're quite lacy-like, and you have to become familiar with each species really to tell exactly what division they're in. The browns, on the other hand, usually include all the kelps, are kind of an olive green color. And over here are some very typical brown. This one happens to be called sea cabbage. It looks kind of like a cabbage that you might have. And then as we pan out over here, you can see that some of these brown algae, most of them are in the division Phaeophyta. Most of them are kind of olive drab in color, and as you look over there where that water goes out, you see some kind of algae sticking up and hanging on the rocks. Most of those are in this division. So these are the greens, the Chlorophyta, the browns are Phaeophyta, and the reds are Rhodophyta. There's one Rhodophyta, one group of the coralline algae that are quite unusual, and I'd like to point those out. In fact, if you come over here in small groups, just one or two of you, and walk around here, which I certainly invite you to do, it would be much less destructive than coming over with a large group, you'll see these, and you might think that they're lichens, or something that's not living encrusted on the rocks. But this one is very encrusted, and there are other specimens here that are a little bit more lacy-like, but they have a hard shell. In fact, the shell is made out of calcium carbonate. These are Rhodophyta. These are red algae that secrete a calcium carbonate shell around them, and so they're called the coralline algae. In fact, coral atolls are primarily made up of the coralline algae, even though the coral animals or hydroids are given most of the credit for it. So that's just about the basis of the food chain out here as far as the macroalgae are concerned. The greens, the reds, and the browns. The greens are also found in fresh water. The reds and the browns are pretty much confined to the marine environment. So let's examine now the various animal phyla. And we'll start with the porifera, the sponges. And now we're in the extreme low water spring tides on your tidal chart there on your lab desk. Now I'm in kind of a surge channel here. Here are some of the browns that I was talking about. They all have scientific names. This one is Alaria. And this is a red. You can see that it isn't a whole lot different. This is porphyra. And here's another brown, this long lacy one that has these floats on it. This one is called a feather boa after the 1920s feather boas that the gals wore around their necks. But let's now look at some of these bright red encrusting forms that I see here. And I'm talking about here the phylum Porifera. And if we examine them very carefully with the microscope, and here's a little piece that I can pick up, it has a kind of a resiliency, almost like shoe leather, as I poke my finger in. But if I looked at it with a microscope, I'd find that there were lots of pore openings. And in among those pore openings, there were larger pore openings. The larger ones are where the water is coming out of the sponge, and the smaller ones are where the water is coming in. And the way it comes in is that there are cells in here that have flagella, and the flagella beat and cause a current. And so the current comes in the small pores and then comes out the excurrent pore. And while it's in there, these cells feed upon the protists that I captured in the plankton net. Sponges are kind of unusual, because if I put this sponge through a very fine mesh net, a sieve, if you will, and I examined what I got out microscopically, I'd see a whole spread of cells out on the slide. And if I looked at them for a period of time, these cells slowly would coalesce back in and form another sponge. So we say that that really isn't a tissue layer of development. What that is kind of acellular. The cells are independent, although they live in a colony and are quite interacting. These sponges down here are different species. Some of them are bright red like this, some of them are bright yellow, some of them are bright purple, but they all have this sort of a encrusting form, the phylum Porifera. Okay, now I'm down in another surge channel. In fact, this is extreme low water spring tides. The water very seldom moves out of this surge channel, just several times a year. Remember the different kinds of algae I was pointing out, this macroalgae. Here's the brown, and here's some red. In fact, this one that looks green right here is a red in the phylum or division Rhodophyta. Here's that feather boa that I was talking about before. It's a Phaeophyta, some of the macroalgae, and I'll talk more about how they fit into the food webs a little bit later. But right now, let's go on to the second phylum of the animal kingdom, and that's the phylum Cnidaria, the coelenterates, including the jellyfish and the sea anemones, which are well represented down here at Yaquina Head. Look at these green anemones down here at my feet, and you can see that as I touch them, they're kind of sticky. Maybe some of you have experienced that in your childhood or maybe even recently. They feel sticky, and the reason that they feel sticky is because it's not a glue. I'm being shot with hundreds of little harpoons. They have stinging cells. In fact, that's a characteristic of the entire phylum. The phylum Cnidaria all have stinging cells on tentacles. If you looked at them microscopically, what they are is cellular harpoons with a little barb hooked onto a harpoon thread, which they shoot out and they stick into my skin. Not very far, and I can't feel it, except when I pull my finger away, the resistance of those little harpoon threads makes it feel like it's sticky. There are other representatives of this phylum Cnidaria, particularly in the jellyfish and the other kinds of stinging-celled organisms. Another division of living things that are well represented right here at my feet is the division Echinodermata, or the spiny-skinned animals. This sea star, for instance, as I pull him up off the rocks, is a good example of adult radial symmetry. When these starfish reproduce, however, they shed their eggs and their sperm directly into the seawater, surging back and forth. In fact, the fertilized egg then becomes part of the plankton that are fed upon by the filter feeders. But as that fertilized egg matures embryonically, it forms into a little larvae which has bilateral symmetry. And not until it settles down on the rock surface and becomes a tiny little starfish does this radial symmetry exhibit itself. Let's look at one of the unique characteristics of the phylum Echinodermata. If you notice on the back of this starfish, there's a little white spot here. And that's a significant little spot. It's found on all starfish. It's called a sieve plate. And that sieve plate, if we looked at it with a hand lens or a microscope, would look like a sieve. In fact, it has a lot of pores in it. This sieve plate is attached to a canal on the inside that circles the entire starfish, and that canal then is attached to radiant canals that go out into each of the rays. These canals then individually are hooked to the tube feet on the undersurface of the starfish. And you can see if I pull this muscle away, these tube feet are stretching. Notice how the tube feet right here have little ampullae or little suckers on the end. Their favorite food are the mussels, as you can see here. And the way they eat them is quite unique in the animal kingdom. What they do is wrap themselves around a mussel, and then they pump water into the sieve plate, through the circular canal, down the radial canals, and they close the sphincter muscles on the end of the tube feet so that when they pump that water in, these tube feet all extend. Then they latch these tube feet on with their little suction discs onto the shell of the mussel and pump the water out. Can you envision? It's a tremendous hydrologic pressure is put on the muscles of the mussel. Then the starfish can relax. It no longer is expending energy through its muscular system. It's this hydrologic pressure that is working against the closing muscles of the mussel itself, the adductor muscles. Soon the mussel tires, and when it opens up just a little bit, the starfish can extrude its stomach, slithers into the shell of the mussel, and digests it externally. A water vascular system that's unique in this phylum. Other representatives of the phylum Echinodermata don't feed on mussels, but they feed on algae. They're grazing organisms. Here's one representative that's fairly common in the very low water. In fact, this is a small variety. It's the red sea urchin. They also have 5 parts, very much like the star, the sea star that I showed you, but it's hard to see because they have all these spines. Here, right at my feet, is a shell of this sea anemone, and you can see it's divided into 5 parts, and these little bumps that you see here are where these rays have attached at one time. The other species of this particular kind is the purple sea urchin, and you can see it also is very much like this echinoderm here, this red sea urchin. If I turn them over, though, their feeding strategy is entirely different. You see the mouth parts here. It's covered with the flesh. But if you could see them a little bit more clearly, you'd find that not only are the shells divided into segments of 5, but the mouth parts also are divided into segments of 5, so that they form a little mouth that works like this. And as they move around on the algae, their mouth is working like this, and they're actually grazers. So they're herbivores out here, and not predators on mussels. In fact, the first one that saw these mouth parts that were dried out because it looked like a little lantern, the first one that saw it was Aristotle. And so these commonly are referred to as Aristotle's lantern. Okay, so far I've been talking about 3 different animal phyla. The Porifera, remember, they have a loose assemblage of cells that we don't really call a tissue development yet because they're interdependent, but you can isolate them and they can live apart. They aren't functioning like a tissue. Then we saw the coelenterate. If you examine the coelenterate, like you see on the screen now, under a microscope, you'll find that they consist essentially of 2 layers separated by a non-living jelly-like thing called a mesoglia. The stinging cells are only some of the cells that they have in these 2 tissue layers. The coelenterates are all radially symmetrical. They have 2 tissue layers. The echinoderms, on the other hand, although the adults are radially symmetrical, the larvae are bilaterally symmetrical. They have a water vascular system. They have 3 cell layers as they develop as a larvae, continuing on to the adult, so they have 3 actual germ layers or developmental layers. All the rest of the organisms that we'll talk about down here, with the exception of the Porifera and the Coelenterata, or the Cnidaria, have these 3 germ layers, out of which develops all the rest of the organs and structures that you'll see. Now another phylum, and I'm sitting on a lot of them right here on the shore, these are mussels. They're bivalves. They're in the phylum Mollusca. They're all bilaterally symmetrical, as is well seen in this one that I've recently picked up here. It's a chitin. You'll see that it probably doesn't look like more than a piece of old shoe leather, but if I turn him over, you'll find that it actually is a living organism and exhibits all the characteristics of the entire phylum Mollusca. They have a mantle, they have a shell, and they have a foot. And tucked down in here, which I'll show you a little bit later with the muscles, is a series of gills down in this separation. Mollusks generally have shells. In this case, they're quite hidden on the back here, but there's a series of shells that you might be able to see right here. There are a lot of different species. This happens to be the black chitin. They come much larger than this, the gumboot chitin, and they come much smaller than this. Some of them have lovely sculptures on the shells themselves. If we look at a typical mollusk, a bivalve, such as these mussels that I have here on the shore, in fact, here we have represented two different phylum. The mussel itself is a mollusk, and the organisms affixed to them, the acorn barnacles, are arthropods. And I'll come back to the arthropods in just a minute. These mussels